got a meeting, so I've got a hard stop at 1 o'clock. And I'm booked the rest of the day, but if you haven't come in to look at your exam, please do, okay? I know there's some of you that got cut short, or they came in and there's like 7 minutes left, and that's not enough time. But here's my thoughts on checking your exams, okay? When you're checking your exams, we did chapter 1, 2, 4, 13, and 8, right? And it's been really helpful to see what kind of questions are you missing, okay? And more often than not, there's only been a couple of exceptions, it's clumped. They missed a lot here and here, okay? Or they missed a scientist here and forgot structure of, oh, that's better. I think I can yell. Okay, allergy season is just sucking this year, isn't it? Okay, so, some here they forgot, structure or something like that, okay? But it's really helpful. Now, if you come in and you're like, well, I'm not sure what chapters are in, that tells me you don't know your chapters well enough, Okay, because you should be like, oh yeah, that was here, you should know it that well. Okay, so over the weekend I started, can you put your screens down? We're not doing anything with screens right now, okay? Thank you. So over the weekend I've been trying to think, how are some more ways that we can get to increase study skills? Because like I say, and I truly believe this with all my heart, is that nobody fails micro because they're dumb. You're here because you're smart, okay? You're here because you're smart. The reason people don't do well in micro is what? Time. It's time. Time to study, right? And some people, it takes 6 hours to what some can accomplish in 1. That's just the nature of being a human being, right? That's just the nature of things. And so I'm trying to find different ways to help you to study. Now, if you're not putting in the time, I can't help you there, right? I can't do that. It's got to be a priority for you. And for those who are taking 50B, working full-time, the wife and 10 kids, plus working full-time, all of that stuff, okay? People say, oh, you can do everything you want. Yeah, not just all at the same time, okay? So, I'm really trying to come up with some ways that will help you be better and get you prepared, because I know what's coming across the street, okay? All right? And if you're trying to do too much, then you're trying to do too much. You're not superhuman. Nobody is. We're all just humans with mitochondria. That's all we got. Okay? So, what I did on the, oh, it's gonna be a long day. Okay. What I did on the modules is this.
How to study.
Huh?
How to study.
How to study. Good. Yeah, you saw that.
That's good.
Okay, where is it? Study links. Okay, good. All right. I put a 7-day planner that you in Excel. Some of you already have paper planners. I have to have a paper planner. I can't do all electronics for a number of reasons. Okay, but how to make a schedule, reading strategies, note taking, and mind maps. Okay, do you know what a mind map is?
Yeah.
Who know? Talk about a mind map.
Whoa.
Whoa, yeah, no, really, seriously.
Isn't it those things you used to do like in elementary school, middle school, with those big bubbles and like the information?
Yeah, it's just like that. It's just like that. So, and so as you're doing notes today, I know a lot of you are recording and that stuff, but try to make a mind map of what we're doing, because we're covering one topic, which is viruses. Okay, and this is it. So you might want to decide that, okay, I can take notes here, but if you don't do your mind map while we're going over it, try one when you go home and see if it helps you. See if it helps you do that. Plus, our SI for 50A and 50B posted, you saw that, right? What you can do in 5, 10, 15 minutes, yeah. So this is a class where you practice being a great nurse, where you practice sitting beside the time. Because what I've heard from this year's class is that it is brutal. You think this is hard? It is brutal over there, okay? I have students that said, oh, well, I just thought I could work part-time. They had to quit their job. We're 6 weeks into the semester, okay? Oh, I can do this with my kids. Oh, no. No, they can't. They really can't. And I'm not trying to scare you or anything. I want you prepared. Because if you're prepared, then you succeed, right? Okay, so mind map. Okay, so I don't know how you want to do it, but what you do is we're going to start out with viruses today. Viruses, okay? And with a mind map, what do? You move things out from it. Yeah, okay. They didn't teach that in school, but we had dinosaurs, so I don't, that's pretty cool that they taught you that. Okay, so let's take a look at viruses today. And you can start your mind map, and the more colors you have, the better. That's what all of the learning is. And if you watch the video on mind maps, it talks about mind maps and colors, and it has things for both. Okay, so let's look at this. Okay. Do you have your virus information sheet too? Okay, I would have that out as well as we're going through things to help you get
The one you updated, right?
the one I updated, yeah, because it was both the same thing. Okay, so here we go with the viruses. Okay. Let's see if I can do 120. Yep, okay. Oh. Everything's breaking. Okay, this was in 2021 when we were in the middle of the COVID. That lovely C word. Okay, all right. In 2021, our population is about number 5 in the world, number 4 in the world. And yet, what happened? We had the most deaths of any country. Why? Because, there's a lot of ways that we mishandled this thing. But if you look at here, some of these are, wow, that's really shaky today. Some of these are viral diseases. Now, is COVID as high as it was back then? No, but we've seen next strain, so we know that, hey, things are changing. Okay, so could the virus become more virulent? Yes. Could it disappear and be? Well, it's not going to completely disappear. It's endemic at this point. Okay, let's talk about some of the mortality rates for things. Okay, Ebola. What's the mortality rate for Ebola? 50%. Okay, MERS and SARS are related. This is severe acute respiratory syndrome. Okay, and it popped up back in 2009, it was around in 2011, and now we have COVID that's closely related. Okay, this has gone down a little bit from 3.4. Seasonal flu is pretty low, measles is pretty low. The 1918 flu pandemic, 10%. Okay, why was this so deadly? Okay, most influenza viruses tend to be in your upper lungs, okay? What happened with this one? It was in the lower lungs, okay? What population do we see with influenza that's the sickest right now? Old, young, very young, and immunocompromised, okay? This hit people in 17, 18, 19, 20 to age 25. Okay, when you see that segment of the population getting hit the hardest, there's something weird about this virus. And this is, okay, so let's talk about the influenza virus, okay, for a little bit here. I'm going to draw this out. And this isn't in your book, so I would take pretty good notes about this, okay? All right. So let's see what's going on in the world today. So here's, yeah. Okay, there's our planet. Okay. Yeah. Okay. Okay, Asia, Europe, Africa, Australia, and New Zealand. Okay, all right. There's our world today. What?
What happened to Antarctica?
Well, since there's psychos on the station killing everybody, we're not gonna have to work. No, I'm just kidding. Yeah, we're gonna disregard Antarctica. Actually, yeah, that's a really interesting thing. They screen you for both medical fitness and psychological fitness, and apparently something went wrong, huh? You know what happened up there, right?
Oh yeah.
Oh yeah, okay. I'm sure it's all over, but yeah, kind of crazy. So okay, we'll do. And then here's Santa Claus. Okay. All right. So we got kind of that. All right. So, yeah, one of my physicians in my unit was the doctor for Antarctica, and he flew in with the Air Force. And he gave a talk at Stanford where he's based. He's the infectious disease chairman for Stanford. And he gave a talk about all the infectious disease that you see up in Antarctica in this isolated population. Okay, and so him and his wife, who's also on the staff of Stanford, on the faculty, did a little study, PCR study, to see how people were exchanging microbes while they're in isolation like this. And one of the things, so he showed this at a talk at Ground Rounds at Stanford, and one of the there was really interesting microbial genetics going on with all the people there, because you're stuck up there for months, right? You can't get a flight in or out, okay? So they had a couple of PEs. What are PEs? Oh, man. What are PEs? Pulmonary embolisms? Okay, why? Because the flight up there is so long to get up there, right? And it's usually Air Force pilots, and they're sitting for a long time. And post-COVID, we're seeing a lot more PEs in general, okay? The second thing that I saw that people asked about was, the hypothermia and hypothermal injuries. And then he had one case of a penis fracture that had to be, and I swear to God, he had more questions about that than he did any of the other interesting microbial things. So it's pretty funny. Not really. I mean, poor guy had to be medevaced out. So, yeah, that's expensive. And yeah, oh, why are you on this medevac? Sorry, broke my penis. Yeah, I'll never say that. Okay, so, all right. So right now, what are we? We're in springtime, right? That's because we got Benadryl on. What's happening in Australia right now? It's what? It's winter, right? Hi, how are you? Good. Yeah. Yeah. See, what is this noise? Do you know?
Oh, this is air conditioning vents.
This is horrible.
Yeah, that's our project facility. Put in a takeaway facility, but that's the air conditioner, right?
Yeah, and
That's the air
it
conditioning.
shakes
It
this.
shakes this. Yep.
Yeah.
And all the projector. Yep.
Oh.
That's from the air conditioning.
Oh my gosh.
Because they have it on now. Yeah. So that's that. But this guy's probably just out of juice.
Exactly. So, yeah. Those were the spares.
Oh yeah.
Yeah, they're dead.
They're dead.
Yeah, they lasted for 5 seconds.
No. Yeah. Sometimes lately batteries are not what they used to be.
No,
They used to make them
no,
to
no.
last. Now they make them to throw away so quick.
Yeah,
And
some
it's
more money,
it's
right? And more expensive.
Yep. So
Yeah.
then buy more. 100%.
Yeah, so that's it. That's the
air
HVAC.
That's the HVAC. Yep.
Oh my gosh.
Yeah, it's an issue in rooms, you know. It's an issue. Let's see. Do you have the other one off? There's only
Yes.
one at a time.
Yes.
Good, good.
Yeah.
Turn that off. Oh, it's already. Oh. Oh.
Oh, that's much better.
Yay.
Oh, thank you.
And here's a gift, the replacement.
Oh, thank you. Okay,
Yeah.
cool. All right.
Yeah.
Thank you so much.
Awesome. Oh, let me take these.
Oh, you
Let
need
me
those?
recycle.
Okay. Oh, you
yeah,
need to recycle?
them. Yeah, unless you have a recycle thing.
Yeah, we do.
You do? Perfect.
I'll take it.
Thank you.
Okay, thank you. All right, that's much better. Okay, so what season is it in Australia? Winter. Okay. So what has happened in the last month is usually what happens is that you have to predict which strains you're going to put in the influenza vaccine. Okay. So H1N1 is always going to be one of them, okay? Why? Because we had a global pandemic with that in 2009, nothing like COVID, but we did. And then they usually put H2N3 or something like that. We're looking at bird flu, which is H5N1, okay? And what happens is they meet, we see what's going on in the world in winter, and most of our new viruses that we see come out of here and Asia, okay? China and Southeast Asia, okay? Why? Because it's about how you live with animals. The influenzas that were the most dead, well, in general, they have 3 types of genes. They have human genes, they have pig genes, and they have duck genes, okay? And we pass genes in influenza back and forth, back and forth, back and forth, okay? Right now, bird flu comes primarily from where? The birds, right? That's where bird flu comes from, is birds. And so it has a lot of bird genes in there. The pandemic of 1918 had more duck genes than most viral things that we see, more than H1N1, okay? And we have to look at, you know, when you're doing epidemiology, you look at the whole thing, what is it? So we have to look at farming, okay? And so you'll say, well, I don't really care about farming. You should, okay? Let's talk about the bird flu pandemic right now. Where are most of the cases? In California. Yeah, they're in California. Where are most of the infected herds? In California. Now, what's the reservoir for bird flu, for H5N1? Cow udders. Okay, that's where the virus replicates, is in cow udders. And so if you're going to transport cattle across state lines, they have to be surveyed by the USDA for bird flu, okay? Now, since there's been massive cuts to USDA, they should be surveying who else? Not just the cattle, but who? The farm workers, yeah. The ones that have contact, especially on these large dairy farms, right? That's who it should be. I mean, we know it comes from cow udders. How many deaths have we had in the U.S. from bird flu so far? Just one, and one in Louisiana, and putatively one in California that's being checked out. But there are a lot of comorbidities with it right now. Okay? Now, one of the things that we look at is, you know, especially why are they arising from here? Because of how they do farming. Okay? When you do farming where you're in close proximity to birds and you're in close proximity to pigs, which are very much like humans, in terms of tissue, now you're thinking, wow, that doesn't sound too good. But in terms of tissue type and things like that, remember we can do pig valve transplants and stuff like that, and we have to be very careful because we can transmit, all right? But we have to look at, you know, we're swapping viruses all the time. Most of the viruses that we're going to be swapping aren't going to be, you know, causing us problems, but, you know, we do have some concerns here. So when we look at farming here, you know, the pigs and the ducks are living underneath the homes that are higher up off the ground with humans. And when you've got that much living together, you're going to pass viruses back and forth, okay? So that's why they're saying, make sure the milk that you're drinking is not raw. Make sure that your pet food, there have been many cats that have died from bird flu. Why? Because people are buying their pets organic. Oh, I want it to be raw and all this stuff. No, you don't. It kills the cats. Okay? So, yeah, there's a lot of things to think about. So, you know, so what usually happens in February of every year is that the world gets together and uses that next strain stuff that we saw, plus other databases, to predict in the next year what's going into the influenza vaccine. And most of you who got the influenza vaccine got a quadrivalent. So what does that tell you? It's quadrivalent. There's 4 strains. So H1N1 is in, usually H2N3, okay? But what happened? The United States did not participate in selecting, okay? As we see how we culture viruses, we can't culture viruses for vaccines on the spot. It takes weeks and months to do that, okay? We missed a window of opportunity, okay? So what is my prediction? You know, if we're not in the game to find out what it is and we miss out, is the flu vaccine next fall going to be very effective? No. And that's my prediction, is next fall it's not going to be as good. This year was 30 to 40%. I'll take that. But if we miss it completely, we're in trouble. Okay? All right? So that's why, you know, okay? And I'll tell you why I'm terrified that we've got such one of the worst influenzas that we've had since the 1990, along with bird flu. I think we got a problem. So anyway, does this make sense? Okay, we talked a lot about vaccines, vaccine selection, all that good stuff. Okay, so let's look at the sizes of viruses. When you look at the relative size of viruses, the adenovirus, which is really tiny, and rhinoviruses, these are cold viruses, okay? And rabies virus is a little bigger. Ebola is the monster of them all. That is a huge virus, okay? Poliovirus, very small, okay? And here's an E. coli in comparison, okay? And here's just a section of the human red blood cell, okay? All right, now we're gonna see some new terminology here. Bacteriophage, bacteriophage. What is a bacteriophage?
Virus that infects bacteria.
Yeah, it's a virus that infects bacteria. Don't confuse the bacteriophages with the phages that infect us, because you're going to hear the term phage used for all viruses, okay? But make sure you know whether you're talking about a phage that infects humans or a phage that infects bacteria, okay? Good so far? Okay. All right, here's some morphology. You can, look at them. When you think of a virus, think of 2 things: genetic material and protein. And the protein, what is the word that describes a protein coat? A capsid. Yes, a capsid. And we got lots of different shapes that we can have of those capsids. Okay, how do we classify? For this microclass, we are going to use a genetic classification system, okay? And if you go down that rabbit hole, you can see there's a genetic classification system, there's a morphological classification system, there's other classification systems based on certain sequences of the proteins, blah, 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 blah. We're going to stay here with genetics, okay? So, single-stranded, double-stranded, all right, double-stranded RNA, single-stranded RNA. COVID is, of course, a single-stranded RNA, okay? So, will you have to know the Baltimore classification system? I'm not going to ask you to reproduce this, but you should be very familiar with it and know which viruses are which, okay? And you can get that information off your virus sheet and also the disease card sheet, right? So you have to know the viruses for that. Okay, viruses tend to be very host specific. Okay, why? It's all about attachment. How does that virus attach? You never get a cold in your toe. Why? There's no cell receptor on your epithelial cells for your toe that give you a virus. But where are the receptors for a cold virus? Upper respiratory, usually upper respiratory, okay? All right, and you have these host receptors are different in different species, okay? What was the receptor for COVID-19? We talked about it last time. What was the receptor for COVID-19? What? Yes, very good. It was ACE2. Yeah, where do you have ACE2 receptors? You have it in your lungs, in your brain, in your muscles, in your GI tract, okay? You have them everywhere. What a target. That's why you get so many diverse symptoms with COVID, is because those receptors are everywhere and everybody's different. Okay? So, there are a couple of ways that you can attach to a host cell membrane. One is for the envelope viruses, they use spikes. That's what these are. These are spikes. Okay? H and N, neuroraminase, and that's a big one for attachment. Okay? And non-envelope viruses, they use their capsid molecules. So where's the capsid again? On the capsid is a protein coat, so they're going to use their protein coat. Okay, we're lucky. A lot of animal viruses are enveloped. COVID was enveloped. So what does that mean in terms of control of infection?
You just have to block the spikes, right?
No, even easier than that. As soon as you see envelope, what should you be thinking? Soap. Soap. Why? Soap breaks up that envelope and activates a virus. That's why, hand sanitizer is great, but what's better for COVID? Yeah, make sure that you're washing your hands with soap and water, because that soap, you see what that Dawn detergent does in your kitchen, right? It's scrubby, right? It gets rid of everything. Okay, well, it gets rid of the envelope viruses really well. Okay, so not just water. Always use soap. Okay, if you can. Okay, let's talk about how we name these things. Okay, again, if they can't decide on how to classify them, they're going to have trouble on how they name them too. So usually you see order, family, genus. Okay, and then we see species. When you talk about the species, we use the common names. Okay, like SARS, severe acute respiratory syndrome. Okay. The related coronavirus, that was SARS-CoV-2. That's where the name comes from. Okay, and when you designate a strain, you see a number. Okay, now sometimes if you want to look up in your free time what the virus is, what was in the quadrivalent vaccine this year, you're going to see numbers. Okay, and why? Sometimes they name the viruses based on where they're found, sometimes the year they appear, okay? But you'll see a number. This is coronavirus 2 because it's number 2 in the series, okay? So this all on this side of the slide shows you the naming for coronavirus. This tells you the genus, the species, and the strain. Okay, so is that cool? You do your cards. All right. Let's talk about cultivation methods, okay? One of the things that we talked about back in chapter 1 was Koch's postulates. Can every disease be, can we use Koch's postulates? No. How come?
Not every pathogen can be.
What? Okay, viruses are hard to culture, okay? It takes a long time. There's a disease card right under your chair. Right there. You're going to miss it. It's got color on it. Okay. So yeah, you can't always culture viruses. So we're going to talk about some of the ways that we can culture viruses. Okay? And one of them is called the viral plaque method. Okay? Now, what happens is we grow what's called a lawn. So on that petri dish, what we do is here's the petri dish, and we grow the bacteria. So it makes a single monoculture lawn, okay? Okay, call it a lawn, all right? And, bacteria and healthy mammalian cells are cool because they have this thing that you probably learned about already called contact inhibition, okay? Especially with mammalian cells, when you're trying to do mammalian cell culture. Okay, once they touch their neighbor, they stop growing, okay? We use that when we're trying to culture things, okay? There are 2 exceptions to this. Cytopathic effects that you see from viral infection and cancer. Cancer cells, they'll start growing on top of each other. and they make these things called foci. Yeah, so most of the time we've got this nice little lawn. This is bacteria, this is mammalian cells, but bacteria do the same thing. They like to grow nice and even. Have you seen that in your micro lab? A lawn of bacteria? You saw with the disc diffusion test, we grow a lawn, okay? All right, now what do? We put the viruses on here, and everywhere that you see is clear is viral cells that died due to a viral infection. That's what you see here. So we are able to culture this because we know the virus, it's killing these cells. We're able to recover that. Okay, and you can see, some of the plaques that we got from different diseases. Okay, here's the poliovirus, and here's Western equine. Okay, different plaques. But that's one way of cultivating viruses. Let's talk about another way. Okay, I want to make sure that everybody's cool on this because I got friends that are emailing me asking me if I ever worked with transgender animals. And I was like, what? Yeah, in your research, do you use transgender animals? And I was like, my brain was very, transgenic, okay? Transgenic animals, yes, okay? Have I used transgenic animals? Yes, we do, for research all the time. Okay, what are transgenic animals? Yeah, they're mice, they're they're rats, but what does transgenic mean? Yeah?
They have genes from other animals.
Yeah, they got genes from other animals in there, okay? And so we use them a lot in immunology for sure, okay? But that's what a transgenic animal is, and so we've got some superhero here. But this is a tumor on a mouse that has has had its immune system wiped out. So we're looking at tumor development here. But we're able to culture a lot of viruses in animals, okay? We don't always have to use a transgenic, but we do use animals. Yeah. Okay, now let's talk about tissue culture. Okay, tissue. Oh my goodness. I'm writing that email as soon as I get back in my office. Okay, so tissue culture methods. This is really a great way. Okay, so let me introduce you to two terms I want to make sure you know. When you see in vitro and in vivo. Okay, what is the difference between in vitro and in vivo? Actually, this is EN, if we're going to do it right. What is the difference?
Vitro's in like the lab, right?
Vitro's in the lab in a test tube. Vivo is
In a human.
in an organism. If we're doing tissue culture, is it in vitro or in vivo? It's in vitro. Yeah. You've heard of in vitro fertilization. What do? Fertilize eggs in lab, and then they transfer. Okay? So, this is the Syrian golden hamster cells, BHK21. They're a very famous line. They come from the kidney line, kidney cells. Okay? These are fibroblast cells. They are good. Fibroblasts are what lay down your epithelial cells. Okay, the blast means they're immature. Okay, and they're good for culturing respiratory viruses like adenoviruses. Adenoviruses are one of the ones that cause the cold. Okay, but we have tissue culture methods. Okay, so now what do we look for? We look for things called cytopathic effects. Okay, look, here's a nice lawn of cells, okay? And then this is post-infection with COVID, okay? All right? Cytopathic effects, CPEs. These are things that we look for under the electron microscope or under the light microscope to indicate that this cell has a viral infection. That's really what we're trying to show here, okay? And this is from measles. Does this look like a normal cell? Okay? To me it doesn't, because why? Suddenly you've got all these nuclei and this giant cytoplasm. That's not right, that's not normal. Okay, can you see down here for herpes simplex? Do these cells look normal? No, no, look at all those nuclei. Okay, so yeah, we look for signs that something isn't right. And it's really obvious here, what happened here in these cells. These look pretty normal to me. They all have, and look, they died off, okay? So that's one of the clues that we look for, are cytopathic effects. So in your reading, they talk about different cytopathic effects. So take a look at that. All right, now we're back to talking about cultivation, because we usually see the cytopathic effects in tissue culture. When we're looking at cultivation, okay, we use rat and chicken embryo cells, okay? They're called, here's some more cytopathic effects. The normal chicken, and here's the Rous sarcoma virus. Here's a normal rat. Here's a Rous sarcoma virus. Okay, you can see big differences between these, all right? Again, more cytopathic effects that are indicating what? That the cells have had some kind of viral infection. Okay, cultivation. This is more cultivation. Embryonic eggs, okay? This is how we develop our vaccines that we talked about earlier, is they're cultivated oftentimes in eggs. Now, other countries use other ways, but we're really fond of this way. That's why you have to sign an allergy waiver when you get an immunization for influenza, right? They ask you, are you allergic to eggs? Because why? They grew it in here, okay? And yeah, so what they do is they take 10-day-old eggs, they'll inject the virus in, and they'll grow it up, and then they'll isolate it. I have a video, question.
If someone is allergic to an egg and like say it's like the necessary vaccination,
They don't get it. Yeah, that's why, I mean, how do you want to be wrong here? Anaphylaxis, they may or may not come out of, or vaccination, where there's a lot of things you can mitigate your infectious risk, so, yeah. Yep, yeah, it happens, yeah. I can't hear you. Influenza vaccine, yeah, comes from eggs, okay? Yeah. So, yeah, so there's a big process. I'll have to try and find the video that shows what happens in the egg process so you can see how long it takes. And it all has to be done sterilely. It's not like chickens from the farm, right? Everything is raised sterilely, okay? So, okay. All right. Okay, the next thing we're going to talk about, so we've talked about cultivation. So those of you who are doing a mind map, you came up with lots of different ways that we cultivate, right? And we look for cytopathic effects. That's how okay, good. So now we're going to kind of shift gears and talk about the T-even phages, which are T4, T2, T6, T12. They're even numbered, okay? That's why they're called T-even phages. That's kind of the general name for them. And if you notice, I think they look really cool, okay? So here's the genetic material, here's the capsid, the protein capsid, okay? And it's got like tail fibers that help it dock. And then what happens? See this pin and plate in the tail sheath that acts like a syringe and injects DNA into the bacterium. So when you see T-even phages, these are bacteriophages, okay? These are bacteriophages, okay? All right, so let's draw what happens in the infection of a bacteriophage. Okay, so I'm going to put our friend E. coli up here. Tell me what you know about E. coli. Negative or positive? Gram negative or positive? Gram negative, motile or non-motile? Motile, right? Okay, except for ours. I don't know what's wrong with ours. We ordered some new ones, so yeah. Okay. What's the arrangement of their flagella? What is it? Amphi. Okay, remind us what amphi is. There we go. Yeah, it's not lophotrocious, it's amphitrocious all over, right? And they do the tumble and run. Yay! Okay, good. All right, so we're gonna have this invaded by a T phage. Okay, what's the structure? The capsid, and then here is genetic material, and it's got this, and it's got the pin and the tail. Okay. Now, what happens is this will dock, and the genetic material will go inside of the bacterium. And here is all of the information to make more protein, assembly of the protein coats, and more genetic information that can be assembled into here. So if we fast forward a little while, okay, this cell has basically been hijacked. Okay, what's going to happen? It's going to make more phages in here. I know my art sucks, sorry. Okay, it makes more and more phages. Okay, all right. Now, let me ask you something about quality control. Is there any? No, get them made and get them out as soon as possible, right? So are you going to see mutations? Absolutely, you're going to see mutations. And there's not time to repair, nothing. They just package them in the heads, and eventually this gets so large, what happens? Okay, there's a word for pop that we use. What is it? Lyse. Yeah, lyses out, and then these go all over and infect other cells. Okay? Got that? Is that cool? Okay. So, this is called the virulent phage. Virulent means that it's going to infect others. Okay? That's going to be different than the phage that we have. What's the other phage that's non-virulent? What is it called? We have. No? Okay, we'll come to it. All right, here's a virulent phage. This means it's going to go out and cause disease. Okay? Now it's pretty amazing. It's not one phage per bacteria, but hundreds. Let's show you a picture of this. These are all artist rendition. That's more like it. Okay. All right. It's not like this. This is nice and clean. No, it's messy. Okay. And you got lots of competition here for assemblage of these bacteria. Isn't that cool? There's another picture. Yeah. Yeah, that's more like it. Yeah, many. Okay. Yeah.
Okay, they're competing against each other to get inside.
To get inside, to get their capsid made, to get assembled. Yeah.
They just keep going until no more can fit.
That's right. Yeah. Yep. It's brutal out there. Yeah, totally. Okay. No. Okay. Okay. All right. So now we're going to talk about the 5 stages of lytic replication. Lytic, so when you look at viruses, they can be lytic, which is what we just saw where they go out and they're going to infect others, or they can be what's called lysogenic. Okay, those are the 2 flavors that they come in. Lysogenic, they're much more stable, they integrate in the host chromosome. We'll talk about those in just a minute. Okay, the first thing is attachment, and this is done by random collision. And when you look at this, the structures here, the tail plate, the base plate, help to attach it to the host receptor proteins. and it's specific. So for example, E. coli doesn't have all sorts of T-even phages. It only has T4, okay? So the one I drew on just a minute ago is T4. Okay, 5 stages here. Stage number 2 is penetration, okay? So you gotta get this information past the cell wall and the plasma membrane. So what is injected is what's called a hydrolytic enzyme, which goes in and helps digest and make the way. And the tail sheath actually acts as a syringe, okay? There's actually little prongs on the end here, and it acts as a syringe, okay? So, and it injects that material once those enzymes have made a way for it to do so, okay? Now, for animal viruses, there's a little bit of different strategy. They can, yeah,
What happens when they inject DNA? Where do they go?
where does what go?
The T-phage.
It stays attached.
It stays attached.
Yeah, it stays attached until the lysis open. Like we saw in the picture, you can see at the bottom of that cell, you're starting to get lysis and there's still phages hanging on. Yeah. Yeah, because really what are they? They're a vehicle to transmit genetic material. That's really all they are. They're not living, so that's all they do. Good question. Okay, so endocytosis, these are for animal viruses. We've talked about endocytosis. Here's the receptor, and it fuses and it either takes in the entire virus or it can take in just the genetic material. It depends, okay? Non-envelope viruses, like naked viruses that don't have that nice envelope that blend nicely with the cell membrane, they have to, the viral genome goes in, but everything else remains on the outside. Okay. All right. So this just kind of shows you the attachment, endocytosis, penetration, and uncoating. Is this an enveloped or non-enveloped virus? It's got to be enveloped because it fused with the membrane, right? These non-enveloped viruses that just have protein on it, they can't fuse with the membrane like this. Okay, so let's look at biosynthesis. Okay, biosynthesis, you're making new things. This is where everything is getting replicated and you're making new DNA with mistakes. Okay, so this is total takeover. All the metabolism that bacterium is doing at that time is dedicated to the virus. That's it. It's stuck. It's over with. Okay, this is a battle lost. Okay, and then we get what's called stage 4, which is assembly and maturation. Okay, so all these pieces parts are reassembled. Okay, and then the cell lyses and they're released. Okay. Yeah, this is so true. We don't understand exactly how phages are assembled inside the host bacterial cell. We don't understand how are they able to do it. Is it just diffusing and meeting up at the right places, or are there chemical markers on there? You know, that's a $60 million question, right? Okay, then we have release, okay? And there's a couple of ways. Oftentimes the virus uses a lysozyme to degrade the cell membrane and cell wall so you can release these, okay? And that's it. So on your mind map that you're making, for those of you who are doing mind maps, yeah, so we've gone through the replication cycle. Okay, let's add some more terminology before we move completely away from this. This is, there's things called eclipse time and burst time. Eclipse time is the time 0 to pre-assembly. Here's the eclipse time. And burst time is 0 to the release of all of this by lysis, and this is the burst size. Okay, so 2 definitions to know: eclipse time and burst time. Okay, is that good?
Yeah.
Okay. Let's talk about how viruses, exit of viruses, all right? If everything's going well for the virus, it's gonna go, it's released, and those viruses go on and infect other cells, okay? That's the lytic cycle. The lysogenic cycle, this is actually very gentle to the host cell overall, okay? Because it goes through a latent period, okay? So what we're going to see and what you should think about, remember when you took Bio 1 and you took anatomy and they talked about introns and exons in your DNA? Do you remember that? And they used to say, oh, those introns, they're just junk DNA. They're not, okay? They're not. Some of that DNA is old viral infections that your ancestors had. You're walking around right now with your genome full of lysogenic viruses. Some that you picked up and some that might have been in your DNA for thousands of years. Okay? Isn't that amazing? You have a history of a lot of viral infections that your ancestors had. Okay? And so what cycle? Well, it can't be the lytic cycle, it's the lysogenic cycle. Okay? So what happens? That you get a prophage. Okay? That's an important new word, a prophage. It remains in the host cell. Okay? Now, temperate phages, they don't get replicated immediately, but the DNA gets incorporated into the host cell. One virus that does this is HIV. Okay, HIV can incorporate into your host chromosomes. All right. One of the signs that we look for when we're treating HIV is a viral load, and there's some new medications out that are fantastic. They get the viral load almost to non-detectable, but is it ever completely gone? No. What's another virus that can go latent and then come back? What? Herpes. Yeah, herpes can go hang out for a while and then come back. Right. Think about the relationship between chickenpox. Is chickenpox ever completely gone? No. Where does it hide out? What kind of cells does chickenpox hide out in? Well, let's go back. What does it come back as? Shingles, okay? And it's usually in the trunk area, but not always. I've seen it in the eyes and the ears before, and that's horrible. That is absolutely horrible. I've seen it on the face, along here. This should be a hint. Where does it hang out? Nerves, right? Nerves. That's where it hangs out. And can the immune system detect it if the virus is hanging out inside the nerves? No. So what do? We give immunizations to train your immune system, get your helper T cells up, your helper B cells up, so when it does emerge, you can make antibodies very quickly, okay? So we're gonna see a lot of these, okay? Now, also, as part of the lysogenic cycle, these bacteria get useful genes from the viruses, like toxins and enzymes and drug resistance. Sounds like an aircraft taking off all the time. Okay. All right. It really is able to do that. And here's some lysogenic conversion. Okay. Normal E. coli doesn't make us really sick, but this one, O157H7, has converted. What? It makes a lot of toxins that you don't see in normal E. coli. Okay, it has this shigotoxin gene. Tetanus, botulism, cholera, salmonella, strep infections have lysogenic conversions. Not everybody gets scarlet fever from a strep infection. What has to happen? Okay, that should be on your cards, right? That should be on your cards. Hopefully it is, right? Because what happens? Not everybody gets scarlet fever, but if there's been a lysogenic conversion, yes, then that person can get scarlet fever, okay? So we've got a new way to transfer genes, transduction. We've looked at pili, right? What are pili? Chapter 4. Okay, those are structures that you transfer genes on. We can also use what's called transduction to transfer from virus to host, okay? All right. So this kind of compares the lytic and the lysogenic cycle. Okay, so we've gone through the lytic cycle. Over here is the lysogenic cycle. Okay, so let's pretend, here's where the phage came in, inserted its DNA, and what is the name of this? It's not a plasmid when it's a viral thing. What is it called? We just looked at it. It's called a prophage. Remember the prophage? Okay, so it's got a prophage in there, and it can go either lytic or lysogenic, and here it goes. So it decides to go lysogenic. So what happens? This gets incorporated into the host chromosome, it gets replicated with binary fission, and it can go for 5 divisions, 10 divisions, 100 divisions, but eventually it will come out. And what happens is it makes a nice little figure 8, and there's enzymes that come and cut it, and now it can go back here and become lytic. Isn't that scary? Yeah. I mean, it's really elegant. These viruses have been around for millions of years. They're going to continue to be here. So, so make sure you know the difference between the lytic and the lysogenic cycle. Okay. All right. So let's talk about how some of these viruses exit. Okay, we've got envelope viruses versus non-envelope viruses. Okay. This is exocytosis and endocytosis, where you know, the material comes in, and then you've got envelope viruses that take advantage of the host cell membrane. That's really creepy, because if you got an envelope virus like COVID-19 from someone else, that virus is enveloped. You've got somebody's plasma membrane from whoever gave you COVID. Yum! Right? Isn't that cool? Yeah, every time, so you're getting ready, you're sneezing and coughing, and those viruses are there irritating your URI, upper respiratory infection, all those cells are there, and the viruses are irritating them, and you sneeze and cough, all of the viruses that you released have part of your cell membranes around them. Yeah, cool, big times. Okay. All right, so we've talked about a lot of the latency, persistent of animal viruses. We talked about latency. Here's some examples. You got them, herpes simplex. Okay, persistent, continuously released. Okay, we talked about how HIV incorporates. All right, so once you have Hep B, HPV, HIV, are you ever free from the infection? No, because you're always releasing a few viruses because it's incorporated. Okay, so that's cool. All right.
Can animals get those?
What?
Can animals get those? Can animals get the persistent ones?
Yeah, they can get persistent ones. Yeah. There's a whole bunch of others. Epstein-Barr is, 97% of us have been, it's herpes simplex 3. You'll learn that on your cards. Okay? Most of us have that. Some people don't. Okay? We know it as mononucleosis, right? It's kissing disease. Okay? Once you got it, does it ever go away? No. It hangs out. It's latent, and it will come out some other time. Okay. So, how do we prevent this? Good hygiene. Okay? Contaminated food, water. I mean, polio. What's the route of polio? Oral fecal. Okay? And so that's a virus we like to prevent. Vaccines. I just saw some new data on the HPV vaccine. So there's like 30 different HPV viruses, and that vaccine I think covers 9 of the ones that can cause cancer. Because viruses cause cancer, you know that, right? Okay? They're seeing a 98% reduction in cancer rates for cervical cancer. That's absolutely phenomenal, okay? That is really, really good that it's able to do that. So vaccines, what they do, they stimulate the defenses of their body. It's two-pronged, right? You've got the antibodies that stick around for several months, but you've also got what over here that stays around long-term. What are the name of those cells? Come on. We're 6 weeks in. You've heard me talk about these cells all the time. Memory T cells, memory B cells, right? Memory T cells, memory B cells. They stick around for your lifetime, okay? Unfortunately, because of mutation rates and things like that, it's not available for all viruses. Antiviral drugs. These are not antibiotics. These are antivirals. And here's a hint that you're on an antiviral. The drug name is going to end with VIR. Okay? Acyclovir. Antiviral. Okay? So you can look at a drug name, you might not know exactly what it does, but you know that if it's got vir on the end, it's gonna be antiviral. Okay? All right. And usually it's just supportive. It's just supportive for a lot of viruses. That's all we can do. Okay. Let's talk about why this flu season is so critical. Okay? So we have H5N1, N1 circulating, and we have H1N1 circulating plus H2N3. Okay, these are our normal viruses that we see circulating. Okay, this has been the worst influenza season that we've had since the 90s. There's been more cases, we're not out of the woods yet. Okay, now, okay, all right, yeah. Okay, so we've got these viruses going, and how are these viruses transmitted? What's some mode of transmission here? Okay, they're droplets, aerosols, okay, depending on where you're at. Okay, what's a mode of transmission for H5N1 right now? It's got to be direct, okay? We're thinking it's direct. But now, with all of these herds being infected, we're wondering if it's, airborne.
Can it be animal related?
Yeah, absolutely. Because the animals are transmitting them within themselves and the workers are picking it up. So yeah, it could be here. Okay, is H5N1 transmissible person to person yet? No. Do you see where I'm going here? All you need is a patient that's got H1N1 or H3N3 and H5N1, and it doesn't even have to be a symptomatic infection. What's going to happen to the genes of those viruses? They're going to be mixing. Okay? And as soon as it picks up, this virus picks up the airborne transmission here, what's going to happen? We got a big problem. We're cooked. Yep. Yep, we are totally cooked. So, are there vaccines being developed? Yes, but what do we use to make vaccines most of the time? Eggs. So, can it be done quickly? No.
Not right now.
Not right now. So, do you see why I don't sleep very much? You're not sleeping very much for a different reason, right? Because you're trying to learn all this, and I'm thinking, oh my gosh, what is going to happen here? Okay, so cooly. All right, so we are done with viruses. Okay, let me turn on the lights. Okay, I'll see you on Wednesday. Just a please for a note,